嗯，
对我们panelist 还有这个何阳刚才 mention 了徐勇的年轻有为，还有安宇，
啊，
安宇是 Philip Sharp 的那个博后，然后也是2年跟我一起开始的2年半之前。安宇，你打个招呼来，也是做
哎，
也
陈宇老师，
哎，你好，你好，
哎，
你好，
我在
张院长你好。
我在国内读博的时候就听过您的这个名字，所以感觉对，就是已经出名很久，然后这是第一次感觉见到您，对，
是吧？特别
久仰大名。
真的真的是对，
你们都
我
是真的就是年轻有为，现在就是羡慕羡慕。
没有都
何阳，
像都
何阳，要不自己介绍一下？
我就想说一下，可能张院长跟我博士导师很熟啊，周玉东认识
哦，
周玉
啊，
东啊，你认不认识啊？以前也是 BI 的。
嗯，
应该是就是跟我 overlap 过吧？
对，
没有 overlap，
没有
偶尔
overlap。
很少，很没有 overlap 过。他以前是 neurology 的，
哦
Cliff Saper 那边的。
哦，那是后来就是那个 Brad 和你们就是
哎，
就是合作最多的那个地
嗯，
方。
对对对。
对 small world，对，
做代
我
谢的，咱们应该就是应该有点可以发生了，代谢
嗯
啊，或者是那个内分泌的
是的，是的。
啊。
是不是要等几分钟是吧？
我差不多
我
8:01，我们要不早点开始？我生怕问题太多，然后搞了，因为青春还在纽约是吧？
我在纽约，我在帮我儿子搬家，所以我们现在在纽约面
哦，
试，找了一个地方。
啊，你儿子都已经就是大学毕业了吧？
今年刚毕业。
对，
这日
那
子过的。
其实你直接从纽约去奥兰多吗？
没有，我要回市再吃，我明天回市。
啊，
嗯
对，所以我感觉问题应该不少，所以就是
嗯
Q&A section，所以会比较长。我们就现在8:02，我们现在开始了，我们差不多也接近80-90个人了。好，我先给个 introduction，Good morning everyone in China and good evening for people in North America. Today is a great honor to invite the Professor Chen Yu Zhang for our metabolic edge talk. Zhang, here is a brief introduction. Zhang received his PhD degree in University of Fukushima and postdoc training in Harvard Medical School. I think they have a lot of friends here, including Qin Chun, Jiande, Qin. He then promoted to assistant professor there. Uh, then he come back to China. Um, Now is a professor in biochemistry department and served as dean of the School of Life Science for Nanjing University. Dr. Zhang made a lot of contribution to many fields with more than 58,000 of citations and have more than 100, actually 114 patents. It's very impressive. Due to time limitation, I only give introduction for one of his pioneer work on RNA biology and metabolism here. I think this is also related to today's topic. Um, he pioneered the field of extracellular RNA and established the concept of extracellular RNA is a mediator for co-evolution and adaptation between species. I think that's not only mouse, human, and also plants. For this summer metabolic talk series, we try our best to balance different topics. I think Dr. is one of them reflect one of the important field for the metabolic field. Also balance the geographic, labs. So Dr. Zhang is the first speaker for this talk series from China, and we also invited, 4 panelists related to his field from 4 American institutions. So I hope this Chinese communication, American scientist communication could inspire some new ideas and advance the science. So, Dr. Zhang, thank you very much for accepting our invitation. Now, stage is yours, please. Yeah, Great talk.
Thank you, Dr. Guan. It's my great pleasure to have the chance to share some my works, and I'm so glad to have the chance to meet a lot of old friends and find the new friends. Today, I will talk about the RNAkines, basically the new another new concept. I hope after my talk, the people is going to use the concept in your research. Okay, and it's okay. Everybody can see the slides, right?
Yes, it's good.
Okay.
Slides.
I changed a little bit the title. Now it's beyond hormones and cytokines. RNAkines are key players in the regional communication. And the regional communication also is a new concept. Comparing the original title, I think it's important to note we have the regional communication. So we will know what's the difference between the RNAkines and the hormones and the cytokines. Today, I will talk about 3 parts. Basically, the first one is extracellular. Okay, the classic biochemical concept is the RNA cannot stably present in the outside of cells. Okay, so for the mRNA messenger RNA vaccine or the RNAi technique, all of them, because of they don't have because of the RNA is not stable outside of the cell, so requires a specific delivery system. While the concept for the RNA unstable outside of the cell is based on the messenger. And around 22 years ago, 21 years ago, I started to study the microRNA, comparing the messenger RNA. The microRNA is small, and a lot of microRNAs and multiple functions. Therefore, we think maybe microRNA is different to the messenger RNA, not only in their own function, but also for the physical and chemical the characters. Then I had the hypothesis, not hypothesis. Let's see, really it's not by accident, but that time I thought the microRNA maybe can stably present in the outside of cells in the body, such as serum and the plasma. Then we used the RT-PCR. The RT-PCR basically a little bit different to the current the people use. We don't use the buffer, just put the serum or the plasma as the buffer to directly did RT, reverse transcriptase, and find they really have the microRNA, not only in the human. Okay, these samples is from me. This is my serum, my plasma. And then my student, the famous student said, well, if my serum or plasma had the microRNA and the other animals also should have, then he checked the rat serum, mouse serum, calf brain serum, fetal brain serum, calf serum, fetal brain serum, and horse serum. They all have. Okay. Then we did characterize the serum microRNA. Basically, they do have different characters to the microRNA in the cells. You will see that if we treat them with RNAs, most of the microRNA in the cells are degraded. However, the serum microRNA basically resistant to the RNA's digestion. In the case, maybe there's a little different. Okay. However, for the RT-PCR, you can detect the signal, but you cannot rule out the possibility this signal is come from the whatever the RNA fragment contamination or even the genome contamination. Therefore, we developed a new method. Basically, this is the first time in the world using the sequencing. We call the Solexa sequencing to check microRNA in the serum. And importantly to note that only using the sequencing technology, we can find the microRNA or the other small RNAs has the intact structure. This is important. Otherwise, if you just check the whatever the fragment, the people say, so what, right? Only we detected the intact sequence of the microRNA that we can propose they have possible biological functions. And then we found 700 million reads and identify 7 classes of the non-coding RNAs. Here, also important to note that we find a lot of tRNA and ribosome RNAs. Before, I think 30 years or 40 years ago, the people already find some the ribosome RNA or tRNA in the blood. That time, people just thought this is just a fragment. It's not the entire, maybe they have a function. Since we find this tRNA and ribosome RNA with kind of small RNA or microRNA structure, then later on, the people also have found that the tRNA and the ribosome RNA also have the mechanism be processed and form the microRNA-like small RNA. And therefore, the tRNA and the ribosome RNA also have microRNA-like functions. I think this is important. This is basically the technique supported. We have found the microRNA or small RNA outside of the cell have intact structure. This is important. I think this is important because if you have the intact structure, it means they possibly they have a biological function. And then we in the blood we also find a lot of different source from the microRNA, like plant microRNA 2911 and the food from the Ebola virus and whatever H5N1 virus derived microRNA, and also we also detected the microRNA from the Salmonella. It means not only in the blood, not only have the endogenous microRNAs, also have exogenous microRNAs. They also plays important role to regulate physiological or pathophysiological condition. Anyway, and also we have found that the small RNA, because of their stability, they exist a lot of many extracellular like the serum, milk, royal jelly, the urine, and the sperm. And this one, we still have not finished the study because we have found the small RNA is present in the reverse transcriptase. We have the hypothesis what's going on with the small RNA in the reverse transcriptase. And importantly to note, not all the branches of the RT, they have the small RNA. Takara RT with a lot of small RNAs. However, RBI, no, the ABIs, they don't have any small. We also know why they don't have, and they shows that the ABIs RT basically they have a lower background. However, they cannot accurately find the microRNA levels. They are show the level normally is 1,000 times lower than the real concentration. Anyway, it means there are a lot of type of microRNAs from the different sources, and also exist a lot of extracellular atmospheres. And then we have, the first microRNA expression profile, the human serum microRNA expression profile. And we also identify the difference of the microRNA profile between the gender and between the age. While we also studied not only our group, or the group, all of the world, they also studied why they are stable. We have found, and with another groups, either way, they carried by the EV, and Tavari and other people also describe some microRNA is binding to the HDL. Also, recently, we have found a lot of microRNAs, they have methylation, acetylation, not only in the plant microRNA, but also in the human endogenous microRNA. Also, they have a sequence dependent, like just before I mentioned the plant microRNA 2911, they have 16 GC amount of 20 sequence. Therefore, somehow they are pretty stable. Okay. Because of the stability, especially, we also find the EV can package these microRNAs. We think the EV secreted or whatever packaged micron A maybe is actively secreted by the cells. Then we have found that when the cell get whatever the cells, all the type of cells have the capability. When the cell gets the stimulation, they selectively package micron A into exosome. This time, they already binding to the argo or risk complex. Therefore, in the EV, not only packaged micron A, but also with the risk complex. Therefore, they can selectively package micron A into exosome, then secreted out of the cells. The secreted micron A packaged in the exosome then delivered into the circulation. Through the circulation, they can send the micron A to the target cells, release the micron A complex binding to the target cells. This also important point here is if we have the complex with the micron A in the targeted cells, then the micron A, exogenous secreted micron A, don't need to binding to the endogenous risk complex in the target cells. Therefore, they have a very low levels of the secreted micron A, then they can have a robust biological functions in the target cells. And we also have found, basically not all group, a lot of group find, you see, intra-tissues, inter-organs, between systems. And you see from the pregnant woman to the fetus, genetics, even between the different species, they all have secreted micron A or extracellular RNA mediates their function. A lot of works right now about this kind of study. I think at least have 6,000, something like this, whatever. They study the micron A's mediates between the organs or whatever the systems. Therefore, we raised the new concept that the secreted micron A is novel class of signaling molecule. They with the classical hormones and the cytokines, they make a new type of secretory regulatory network. One, the normal secretion maintenance of, they can maintain the homeostasis, while if they have pathological or disease, they can secrete pathological micron A's and cause disease. Okay, this kind of extracellular RNA, especially the secretory micron A, is an important signaling molecule to mediate intercellular and interorgan communication. Then why I need to have or name the new concept RNAkines? Okay, the RNAkine basically comparing to the first of all, the secretory microRNA always called the EV RNAs, exomal RNAs, or the secreted microRNAs. This not enough to show the secreted microRNA, the function and the importance. Okay. A only focus on the spatial localization and fail to address the unique selective release and the potent signaling characteristics of intracellular non-coding RNAs. Therefore, we just the beginning of this year, we published a paper they called RNAkine. The RNAkine is a new type of secretory RNAs, comparing to the not only clarify the EV RNAs or exomal RNAs, but also importantly to clarify classic hormones and cytokines. Okay. In my lab meeting, professor from North Carolina heard my story and said, well, maybe it's very good to use the Abraham Lincoln said, government is of the people, by the people, for the people. That I can use this, the hormones, the classic hormones and the cytokines is by the local, of the global, and for the global. While the RNAkines basically is by the local, of the local, and for the region. Why I did not use the local for the local? Basically, the local like endocrine, no, the autocrine or paracrine, and cytokines basically have the function normally, or autocrine and paracrine. This is for the regional, especially mediates inter-organ communication. This is the difference. Okay, I'm not sure you guys get the point or not. Anyway, if you have the question, well, we will going to is it modify or concept because this is another paper we will send it out is the regional regulation, regional communication. For the communication systems and multiple cellular organisms like nerve systems, endocrine signals, inter-organ communication, and the hormones like. Anyway, the point is, if we classify the communication by their effects, we called level 1, global systematic, like hormones or cytokines. By the local, the organ, that is not for of the local, because ask them to secrete is not from the local, is for the global, and for the global regulation. While the level 2 is organ to organ, therefore is by the local, of the local, and for the regional regulation. This kind of I think we define whatever the difference between the hormones and RNAkines. Okay, the hormones, I don't go to get into the detail. Everybody knows, right? And then, since our talk is related to the metabolism, therefore, with the following examples, we not only support, using this example, to support my new concept, but also describe the RNAkines in the glucolipid metabolism, tumor, and inflammation. Probably you guys will, do more, okay, of the RNAkines. Okay, for the glucolipid metabolism, level 1 basically is globally, like insulin, glucagon, leptin, adiponectin, etc. For the pathophysiological levels, they also have whatever the insulin, leptin, and the increased adiponectin levels decreased. Maybe the fatty acid also is the signal. But this is also for the global homeostasis. Okay. For the level 2 is organ by organ. I will have another example to see the story. For example, the beta cell, of course, they will secrete insulin. And in the fasting state, the insulin secretion is down. While we have identified, they also can secrete MIR29s, the family. They can directly get into the liver, hepatocytes, increase gluconeogenesis, and then increase hepatic glucose output. This is also important, right? For the fasting state, you don't have the glucose, and the other tissues, especially the brain, also need the glucose as a fuel. Therefore, the whatever the signal getting to the beta cell, that they use the secretory microRNA to perform or to reach the goal. Let's say this way, to increase the hepatic glucose output. And then for the physiological levels, okay, total exposure, the brown adipose tissues, right? Let's say here. And importantly, we have found cold stress stimulate the brown fat, and the brown fat, of course, will produce heat, right? If you produce heat, you need more fuel. You need more fuels to work, right? Then the brown fat secrete microRNA. They getting into the liver, increase hepatic glucose output. This case is very clear to support my claim that the secretory microRNA is not for the global regulation. It's just for the organ itself purpose, right? It's not for the global glucose homeostasis. I always make this, let's say, like the central government ask Jiangsu province to pay more tax, and using this extra tax to other province, while the Jiangsu province would have the capability, right, to have more tax to the central government, then they have kind of the signaling. For example, they will ask whatever the company ask the Zhejiang province, ask Shanghai to build up the manufacturers, build up the companies, whatever. And then this signal from Jiangsu province and for the Jiangsu province. Okay. And then this is the cold exposure bad thermogenics, ask more fatty acid for the liver via RNAkines. And this is for itself. And a lot of examples. They are exercise for the skeletal muscle, whatever, This is examples. Anyway, this is for itself. Organ to the recipient organ. And another example is the liver. So I'm not getting to the details. This especially in the metabolism or metabolic regulation. This secreted microRNA plays important role, or RNAkines plays an important role, mediating intercellular or interorgan in the organ communication. We call them regional regulation. Okay, this is another examples. And fat, this is also another examples. Fat cells. I'm not sure you guys were not the RNAkines group about 7 years ago or 6 years ago published a paper, and this is true. In the blood, 50% of the exome is come from white fat. And then we demonstrated that 20% of the exome in the blood is come from the hepatocyte. Therefore, what's the difference? This is the exome from the different tissues. They can get into the different recipient organs. They have kind of tissue specificity. Therefore, for example, liver, basically, they cannot directly contact skeletal muscle by the RNAkines because hepatic exosome cannot get into the skeletal muscle. And the fat or liver is barely can pass through the blood-brain barrier, while the white fat or fat exome, they can pass through the blood-brain barrier. Okay. This is just an example. I'm not getting to the details. And then this kind of a summary. This hierarchically, the endocrine regulation, they have a different phase. Before, the people also described some of the cytokines like Bruce described the interleukin-6 secreted from skeletal muscle and getting to the beta cell, right? However, the main regulator of regional regulation has not been studied and has not been classified. And they not occasionally, it seems very obviously the hormones, and even you think about the leptin, the adipokines. Right now, I just reviewed the paper. They call the myokines, the whatever the cytokines secreted from the skeletal muscle. And the Ron published the paper. They also include secrete microRNA in the white fat. They call it adipokines. Well, this is true. We can call this adipokines. How about the other tissues, right? So this is why the RNAkines and their regional regulation is very important. The brain homeostasis, and for the leptin, okay, we of course function in the brain with like Qing Chun did excellent work, and Brad also did excellent work, right? Worked in the brain while is not regulate brain homeostasis, right? They would control global glucose or the lipid metabolism homeostasis. And we be at the Gulou Hospital, and we all group published a paper that the fat cell, obese fat cell, secreted pathogenic microRNAs and pass through the blood-brain barrier caused AD. Okay, and another one is inflammation. Most of the cytokines, why we think the cytokines is control the global inflammatory homeostasis, because they are receptor in all of the tissues. Well, this is kind of evidence. Maybe not enough. Anyway, the cytokine mostly is work for the global regulation. And for the level 2, you know, this also obese adipose tissues derived mere 155, you know, caused colitis, okay. And the inflammation also, you see the level 1, and think about the level 2 is RNAkine. Inflammation, okay. And for the also the kind of inflammation, you see, from the monocyte, mere 55 to the epithelial cells. Okay, this is for the inflammation. And for the tumor, even I don't need to talk more about whether this RNAkines is only for tumor cancer cell survive. Only for. Therefore, the tumor cells secrete whatever the microRNAs or other. Either way, they will affect the immune system, affect the brain. Even Wang Hongyang's group published a paper, I think about 8 years ago. they have found that secreted linked RNA related to the resistance, drug resistance, chemotherapy. Okay. Anyway, the only the tumor, I don't need to clarify what's their purpose of the secretion microRNA or the RNAkines. All the secreted microRNAs is main layer to regulate micro environment for their self or for their survival. Okay, the tumor, I don't basically not need to get into the detail. The summary, this is a typical example for itself tumor. And then I have the conclusion here. RNAkines is key players in regional communication. And I hope, you know, all people in the metabolic field or in the endocrinology, they will appreciate this new concept, and then finally they can use it. Okay, this acknowledgements. It's the famous guy basically help me did the work, find the extracellular RNA, serum RNA, and she is help me writing the paper, regional communication. The concept of regional communication is raised by this guy, and this guy mainly has written the paper of RNAkines. This a new type of a signaling molecule shaping physiology and disease. And the other people also did extremely important works to build up the new field of extracellular RNA. And this is Tony. We also discussed a lot. Finally, thanks for all your attention. Thanks.
Thank you. Thank you so much, Dr. Zhang, for the beautiful talk and the overall big picture in this field. We really learn a lot about the term RNAkine. And kind of fully understand why and how it come from. So we're running the Q&A session now. Let's do the panelist first. So our first panelist is Dr. Qingchun Tong. Dr. Tong.
Hey,
please
it's been a long
go
time,
ahead.
right? So I just asked a question. So, I mean, you guys hear me okay?
Yeah.
Okay. So, it's
Yes, good.
as always, you like always play with novel ideas, novel concepts, right? There's a lot of novel concepts here. So I have two kind of basic questions because you kind of didn't describe a lot of data in detail. Maybe it's kind of clarification. So so you kind of compare this microRNA with the hormones or cytokines. So hormones, we know that can be released, but they always use receptors, right? So cytokines always use receptors. But this microRNA, I mean, I don't know, I mean, they don't have a conventional receptor on the membrane. So you also say they're kind of specific to some kind of organs, not it's like, so can you go a little bit in detail how that specificity, how those microRNA can get into cells. That's the first question. So second question is, especially specifically when you talk about this during fasting, the beta cell secrete microRNA can go to liver to regulate glucose metabolism. So we know that the fasting state, insulin is reduced, secretion is reduced. So we know that the beta cell kind of got inhibited, right? So that caused the inhibition of release. But how that can increase microRNA secretion? So how these two different processes can occur from the same cell, one reduce, one increase? There's two very basic questions because you didn't go in very detail. This kind of clarification questions. Thank you.
Yeah, thank you very much, Qingchun. These very good questions. Basically, you extend our concept. Let's say this way. The first one, the specificity secrete microRNA or RNAkine. Specificity of the RNAkines is in the 3 different levels. First of all, the cell get different stimulation or stress. They can selectively package different microRNA into the vesicle. Okay, for the different purpose, right? The second one, the exosome membrane, they have specific proteins. They kind of like ligand to the receptor, kind of, not exactly the same, to train some specific organ or tissues. We cannot the single organ or tissue specificity, but we can have some, let's say this way, right? Okay, just before I mentioned the liver, they have they can increase the lung, kidney, spleen, pancreas, T cells, but they cannot at all get into the skeletal muscle. Barely into the heart and the brain. For the Y fat, the exome can pass through the blood-brain barrier. And for the stomach, pit cells also secrete, or every type of cells have the capability to secrete, right? They also can pass through the blood-brain barrier. But the pit cell secrete much less exosomes comparing to the Y fat and liver. The second levels of the specificity is the getting to the different recipient cells. They have different targeted genes. Even they say that you are a lot, but different type of cells in the different status, they have different expressions genes, right? And therefore, the 3 levels specificity, let's say this way, basically narrow down a lot the secreted microRNA or RNAkines in the one specific standard to regulate other type of cells. This is the first question. For the second question is, the people also when send the paper to the, 7 years ago, we already sent to the cell and cell metabolism. Anyway, they asked the same question. I said,
I was not the one reviewing it. Okay.
They said, well, we don't know exactly. We don't know exactly the insulin secretion and the microRNA secretion, the regulators of them. We don't know exactly, but we know some of them are similar, some of them are not. For example, the chronic fatty acid, they can increase the microRNA miR29 expression and secretion. The high glucose only increase miR29 expression, but not stimulate their secretion. And for the KCL, both they can stimulate insulin and microRNA secretion. If you think about the type of the secretion, basically the insulin is stored in the granules, right? Then we can infusion to the membrane, then release the insulin. And the exosome is not, is kind of a granule, but it is not the same source of the insulin granule. Therefore, the secretion, the regulation of these two secretions are not same.
Okay, thank you. Yeah, that's
I
it for
just
me.
follow the first question of Dr. Tom. So actually you mentioned some mechanism for maintaining the RNAkine stability. So speaking of the cell specific, the cell specific iron kine. So I wonder if there any way to identify the iron kine from different cell or from specific cells. So that's the from the the donor cell perspective. Because we know that the EV's ion or some kind of exon ion, this is probably could be cell specific and based on the surface marker or the signature. But for other mechanisms, is there any way to distinguish the cell specific ion kind?
Thank you, Xiaoqing. Very good question. Basically, also, we 13 years ago, we sent the paper. They also asked the same question. Basically, we describe the tumor cells secrete microRNA 214 and getting to the T cell and target the P10 and increase the Treg, right? The review asked us how to clarify in the T cell the mere increased elevated 214 is from the tumor cell or is just increase in the T cells, right? The very simple.
Mm-hmm.
If from the exogenous donor, let's say donor cells, they are pre-microRNA and the prime-microRNA would not increase. Then you just check the microRNA, the pre and the prime. You know what I mean?
Mm-hmm. Yes, for the microRNA,
yeah.
for the mRNA.
Yeah, the mature microRNA is the same, right? But for the exogenous,
Mm-hmm.
I mean the donors, they will not increase the prime or pre. Okay, let's see the microRNA. The microRNA produced like this process. They have first in nucleus, they have a prime microRNA, and processed by the JOSA and become the pre-microRNA. Then the pre-microRNA out of the nuclear and go to the plasma, cytoplasma. Binding or whatever they recruit, Argo2 and the dicer process the pre-microRNA into the mature microRNA. The mature microRNA then go to the ribosome, binding to the target genes, messenger RNA, and shut down the block the translation, right? This is a very important question you asked because we have found the exosome or whatever the EV carry the microRNA all are mature. They don't have a pre and a prime. And they also already bonded to the risk complex in the exosome. Therefore, when you find in the whatever the tissues like a liver, a hepatocyte, they have a increased whatever the micron is, then you just want to check. You just check the pri and the pre. If they are endogenous increase the microRNA, the pri and the pre should be increased. Otherwise, this increased mature microRNA is come from different tissues. It's clear?
I see.
Okay.
Thank you. Yes, thank you. And second panelist, Dr. Qin Yang.
Qin.
Hi, Qin, great talk, interesting. You know, I knew a story 20, 30 years ago, but still kind of refreshing, very good. Now, I think Qin asked 2 questions that I wanted to ask. So the great minds think alike, you know, that's the way it is. But just additional question. So the circulating microRNA concentration, so comparing to the cellular levels, is that just 2 copies or 1,000 of copies there potentially would affect the as a hormone, you know, level type of things?
Okay. Your question is whether the such low levels of circulating microRNA or the RNAkines, and why they have the robust biological functions in the receiving cells, right?
Yeah.
Okay. This is the very critical question in the microRNA field. Since the 16 years ago, I found the microRNA or the other small non-coding RNAs is existent in the blood. The whole world thought there could be a biomarker, no problem. While we published the first paper on the 2010, right? And still no. Or maybe 1/3 people in the microRNA or RNA field doesn't believe the secretory or circulating microRNA has the biological function. Okay, we already observed the circulating or the microRNA concentration normally is 1,000 comparing to the classic hormones and the cytokines. Why they can have such, let's say, the robust effect in the recipient cells? For the whole mechanism I think I have no answer. I don't know. While some of them, some of effects like they already binding to the risk complex. Therefore, in the recipient cells, the microRNA don't need to, microRNAs don't need to compete to the endogenous microRNA to binding to the risk complex. We already knew that if the microRNA in the cells, the affinity to the risk complex is about 100 to 1,000 times less than the endogenous microRNA. Therefore, we already, not we, the field already demonstrated that in the one cell, about 100 to 200 copies of microRNAs, they already have enough capability to regulate the the target genes. Okay. And just before, Qingchun also asked, they have different levels of specificity. And we also have another finding, not published yet. This microRNA can be recycled. So, this is just probably the explanation. But of course, I cannot, completely answer your questions.
Right.
The strongest evidence is if my, medicine, the drug, siRNA based on the circulating microRNA work, then the whole field will not question. Even we still don't know the precise mechanism.
Great. Thanks a lot.
Yeah.
All right. Next, Aaron.
Oh, yes. Yeah, Chen Yu, I think it's a fascinating talk. I really learned a lot. I think this novel concept, and also I think I feel very fascinating because I'm working on adipose tissue biology. Actually, you mentioned several times that adipose tissue is very specific, like produce a lot of microRNA into circulation and also can easily penetrate BBB, talking to the brain. So my first question is actually related to this. What do you think is the mechanism about this specificity of adipose tissue derived, like small RNA in exosomes? Uh, uh, does adipose tissue has a like special ability to produce more, or they can easily package the microRNA into exosomes? What are your insights? Yeah, that's my first question.
Okay. Very good question. And we let's say it this way. First of all, we use the liver as the bioincubator to self-assemble RNAi. It's kind of a microRNA secreted, right? As secreted out. And we have found that the hepatic exon barely pass through the blood-brain barrier. And that our collaborator, Bian, at Gulu Hospital, last year published, or 2 years ago, or last year published paper, right? The Y-fat, the diabetic Y-fat secrete microRNA in the EV can pass through the blood-brain barrier, caused AD, kind of, right? And that there are consequent studies is maybe I don't. I will not see the details because they have specific protein in the Y-fat derived EV membrane. Then they cause the Y-fat EV pass through the blood-brain barrier. While in the brain, whether they have a specificity to the glial cells or to the whatever, the neurons, I don't think so. Basically, they already they have two identified proteins, membrane proteins, and another question is basically I think based on if the Y-fat secreted exosome carry out microRNA into brain in the physiological conditions, what's the purpose? And I talked to Biye and Tang Qiqun.
Yeah.
I said this is basically from the evolution. Maybe just the speculation. Maybe in the beginning they don't have the specific purpose, while just have the specific exosome can pass through the blood-brain barrier.